Let me start with a simple but powerful thought. Every tablet we manufacture, every capsule we release, carries something more than just medicine. It carries trust. But what happens when a hidden impurity threatens that trust? This is exactly what happened when nitrosamine impurities were discovered in common medicines. Since then, nitrosamines have become one of the most critical regulatory topics in the pharmaceutical industry. In this presentation, I will walk you through their regulatory limits, the science behind those limits, and what it really means for pharmaceutical professionals like us. So first, what exactly are nitrosamine impurities? Nitrosamines are classified as dangerous genotoxic impurities. They are typically formed when amines react with nitrosating agents during manufacturing or storage. What makes them extremely serious is their classification as cohort of concern carcinogens. This means regulators consider them among the highest risk impurities. Even at very trace levels, they can damage DNA. And that is why their presence is strictly controlled. Not just monitored, but tightly regulated. Now, this brings us to the most important concept: acceptable intake, or AI. This is the scientific foundation of nitrosamine regulation. Acceptable intake defines the maximum amount of impurity a person can consume daily for up to 70 years without significant cancer risk. This limit is based on a very strict safety model. It assumes only one additional cancer case in 100,000 people exposed over a lifetime. These limits are not guesses. They are derived from detailed animal carcinogenicity data and toxicological evaluation. This ensures maximum protection for patients. Now, let's look at the actual regulatory limits. Different nitrosamines have different acceptable intake values. For example, NDMA and NMBA have limits of 96 nanograms per day. These are primarily associated with liver and gastrointestinal toxicity. NDEA and NMPA have even lower limits of 26.5 nanograms per day because they are highly potent carcinogens. Similarly, NIPEA also has a limit of 26.5 nanograms per day due to its strong genotoxic nature. These extremely small limits highlight how dangerous these impurities can be. Now here comes the most practical and important part. Regulatory limits are given in nanograms per day. But in pharmaceutical quality control, we use ppm. So we must convert acceptable intake into ppm using the daily dose. The formula is simple. Limit in ppm equals acceptable intake divided by daily dose. For example, in valsartan, 96 nanograms divided by 320 milligrams gives 0.3 ppm. This value becomes your mandatory quality control specification. This step is critical because this number determines whether your product passes or fails compliance. Today, regulators expect every pharmaceutical company to follow a structured approach. The first step is risk assessment. You must identify whether nitrosamines can form during synthesis or storage. The second step is testing. This involves confirmatory analysis using highly sensitive instruments like LC-MS. The third step is submission. You must update your regulatory dossier and implement control strategies. These steps are not optional. They are mandatory for compliance. Now, in some cases, nitrosamines are even more complex. Certain drugs can form unique nitrosamines derived from the API itself. These are called drug-specific nitrosamines. In such cases, generic limits like 96 nanograms do not apply. Instead, a custom acceptable intake must be calculated. This involves structure-activity relationship analysis and toxicological modeling. This is especially important for new chemical entities, and it requires advanced scientific evaluation. In real-world manufacturing, controlling nitrosamines is challenging. They can originate from many sources, such as solvents, water, and reagents. Sometimes, they can even form during storage and packaging. This creates stability concerns. Detecting them is also difficult, because their levels are extremely low. This requires ultra-high sensitivity analytical instruments. So nitrosamine control is not just about testing. It is about complete process understanding. Today, nitrosamine regulation is a global priority. ICH is developing global standards under M7 Addendum 2. FDA and EMA have issued mandatory nitrosamine guidance. WHO provides global technical reference documents. All agencies are working toward harmonized safety standards. This ensures consistent protection for patients worldwide. And it makes nitrosamine control a permanent part of pharmaceutical regulation. Let me leave you with one final thought. Nitrosamine control is not just regulatory compliance. It is patient safety. Every limit we follow, every test we perform, every specification we set, is ultimately protecting human life. That is why 100% compliance is required. Because even a typical limit, like 0.3 ppm, can make the difference between safety and risk. And that responsibility lies with us.